Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Hi, Assalamualaikum warahmatullahi wabarakatuh. So in this video, we continue with protein synthesis, but we proceed to the second process, which is translation. Okay, so hopefully at the end of this video, you can describe the relationship between base sequence in codon with specific amino acid using genetic code table. Explain translation in protein synthesis. So as we know, protein synthesis involves two processes. The first is transcription that occur in the nucleus, which is using DNA as a template to produce mRNA. After mRNA produced, mRNA will be transported to the cytoplasm to undergo second process, which is translation. So in translation, it may produce protein, or we call polypeptide. So transcription already discussed. So now let's discuss about translation. So before we start, okay, you need to know what is meant by codon. Codon. So basically, codon is a triplet base in the mRNA. Triplet bases in mRNA. We read the triplet base from 5' to 3'. Usually, we read the codon from 5' end to 3' end. And one codon is specific to one amino acid. Okay, one codon specified to one amino acid. So in amino acid, total amino acid is 20. So this 20 amino acid, the codon is about the arrangement of 4 bases in the DNA. So as you know, the base in DNA is adenine, thymine, cytosine, and guanine. And then 4 bases in mRNA, which is adenine, uracil, cytosine, and guanine. So these bases will arrange in a triplet base. So we call it as codon. Okay, basically DNA strand, okay, will use as a template to produce mRNA. So the genetic information from DNA is being transcribed into mRNA. Then I told you the triplet base in mRNA, we call it codon. One codon is specified to one amino acid. For example here, the codon is 5' UGG 3'. So this codon, it will code for amino acid trypsin. Okay. U will code for amino acid penin. Okay. So here the mRNA, so the codon is triplet base. AUG is a codon that code for amino acid methionine. CCC for proline. AGA for arginine. So now you can see the triplet base on mRNA is called codon. One codon specify or code for one amino acid. So this is the characteristic of genetic code of codon. Okay, it must be in triplet bases, almost universal, non-overlapping, means that AUG will code for methionine only, not for serine or not for others amino acid. And then it can be degenerate. Okay, degenerate, or one amino acid can be coded by several codon. Okay, so after this I will show you what it means. Each codon is specific for one amino acid. And then you need to remember this. The start codon is AUG. Okay, the start codon for translation to occur, it must start with codon AUG. And process of translation will be stopped when it reach one of the stop codon. So we have 3 stop codon, which is UAG, UAA, and also UGA. So remember, start codon is AUG, stop codon is UAG, UAA, and UGA. Wak, wa, uga. Right. So this is the genetic code table that shows the codon code the 20 essential amino acid. So one amino acid can be coded by more than one codon. For example, here's serine. Okay, the codon UCU, UCC, UCA, UCG will code for serine. But UCU, you cannot see the combination of UCU in other amino acid other than serine. So please understand, one amino acid can be coded by more than one codon, but one codon will code for one amino acid. Alright, so now let's proceed to the translation process. So this is the overview. Okay, we have done about transcription with the formation of mRNA using information or genetic information from DNA. So in translation, okay, this is the three important tool. Okay, we need to use tRNA, mRNA, and also rRNA. So let's take a look. What is tRNA? So in chapter 1, you already know one of the difference between DNA and mRNA and RNA is DNA will only exist in one basic form. But for RNA, it will exist in three different forms. Okay, tRNA, mRNA, and rRNA. So what is different between these three? Okay, tRNA. Okay, this is the structure of tRNA. So the shape of two-dimensional clover-leaf shape. So here is the amino acid attachment site. 3' here is the amino acid attachment site. Okay, between the base is the hydrogen bond, and this is called the anticodon region. So the two important regions to be known, the site for amino acid, the binding site for amino acid here, the 3' end, and then here at the lower part, okay, is the anticodon region. This anticodon region is complementary to mRNA. If the mRNA is 5', GGA 3', so the anticodon sequence is 3', AAG 5'. So look at here, they are anti-parallel and they are complementary. So the major function of tRNA in translation, it will transfer the amino acid from cytosol to the growing polypeptide in the ribosome. Transfer amino acid from cytosol to a growing polypeptide in a ribosome. So this tRNA, okay, can be used repeatedly. And it will consist a single RNA strand about 80 RNA nucleotide long. And then the tRNA which already bind to amino acid, we call it as amino acyl tRNA. Okay, tRNA that already or bind with the amino acid, we call it as amino acyl tRNA. So the activation or the binding of the amino acid to the tRNA involve enzyme called amino acyl tRNA synthesis. So the binding of amino acid, the tRNA is catalyzed by enzyme amino acyl tRNA synthesis. So here is the animation about the activation of tRNA. This is tRNA, okay, naked tRNA, okay, mean it will not bind to any amino acid. So this is enzyme amino acyl tRNA synthesis. This enzyme will catalyze the binding of amino acid arginine to the tRNA by forming a bond. So now this is called amino acyl tRNA. You get it? Single tRNA. Okay, this is amino acid. So the binding of amino acid to the tRNA catalyzed by enzyme aminoacyl tRNA synthetase. So the binding of amino acid to the tRNA may form a structure called aminoacyl tRNA. Okay. Next, we use in translation is the ribosome. So the function of ribosome here is to facilitate the specific coupling of tRNA anticodon with mRNA codon during protein synthesis. Remember, to facilitate the specific coupling of tRNA anticodon with mRNA codon during protein synthesis. So the ribosome has two important subunits. This is called a small subunit and this is referred to large subunit. So basically here you can see that ribosome has three sites. Okay, A site, P site and E site. A site, this is the site for amino acyl tRNA. Amino acid tRNA means the tRNA which bind to one amino acid. Okay, tRNA that already bind to one amino acid. So the place of amino acid tRNA is at A site. P site is the site for peptidyl tRNA. Peptidyl tRNA means the tRNA carry growing polypeptide. So tRNA carrying growing polypeptides means the tRNA will bind to the more than one amino acid. So if you see the structure of tRNA carry more than one amino acid, so it refers to peptidyl tRNA. So the binding site for peptidyl tRNA is at P site. So E site is for exit site. Okay. Alright. So translation, basically the process whereby the genetic information in mRNA in form of codon is translated into sequence of amino acid to form polypeptide or protein. So process, okay, the information in form of codon in mRNA is translated to produce polypeptide, which a sequence of amino acid. So the process have three stages, same to transcription, but the detailed process is different. So remember, the translation process also have three stages: initiation, elongation and termination. So as you can see in this slide, for elongation, we have three sub-stages: codon recognition, peptide bond formation, and translocation. So let's go through stage by stage. So first is initiation. So here is the mRNA. Then here is the small ribosomal subunit. So the first thing happen in initiation, the small ribosomal subunit bind to mRNA and move along mRNA until it reach the start codon. So start codon is 5' AUG. So here is the mRNA. Small ribosomal subunit bind to mRNA, move along mRNA until reach start codon, which is 5' AUG 3'. Second, the initiator tRNA with anticodon 3' UAC, okay, with complement to the start codon will bind to this, okay, and the initiator tRNA will carry amino acid methionine. Why it carry amino acid methionine? Because start codon, the codon is AUG. So when we refer to codon table, AUG is code for amino acid methionine. So that's why the tRNA carry methionine. So look at here. This is 5 prime. This is 3 prime. So this is called codon because it in mRNA, and this is tRNA. So here is anticodon. They must be complementary. Okay. AU. Okay. A will pairing up with U pairing up with A, G pairing up with C. And this initiated tRNA carry methionine. Next, large ribosomal subunit will bind to the complex, and this initiated tRNA will occupy P site. Okay. See, this is large ribosomal subunit. It may bind to the complex that consists of tRNA, mRNA, and small ribosomal subunit. And then this initiated tRNA will occupy P site. Okay. In elongation, as I have mentioned, it has 3 substages. It starts with codon recognition. What is meant by codon recognition? Okay, the next coming tRNA, it must carry anticodon complementary to the codon at A site. So as you can see here, at A site, the codon is 5 prime, C, 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 3 prime. So the next anticodon must have 3 prime, G, 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 5 prime. And it must carry the amino acid code by codon CCC. So if you refer to the genetic table, CCC will code for amino acid proline. Okay. So now we have 2 amino acid tRNA here. So what happened next is the second substage in elongation, which is peptide bond formation. In this, as you have learned in chapter 1, peptide bond will form between 2 amino acid and involve process condensation. So amino acid methionine may form peptide bond with proline. Okay. So here, this tRNA lost the amino acid, and then the tRNA at A site already have 2 amino acid. As you can remember, as I said, A site is a site for amino acid tRNA. Amino acid tRNA is the tRNA that carry only 1 amino acid. But now the tRNA already have 2 amino acid. So the third substages occur, which is translocation. This tRNA now is called as peptidyl tRNA because this tRNA carry growing polypeptide. So the site for peptidyl tRNA is not A site but at the P site. So translocation means the ribosomal subunit will move 1 codon ahead. Okay, so here look at okay. Now look at this. Okay, this peptidyl tRNA occupy A site. Now the ribosomal subunit move 1 codon ahead. So this peptidyl tRNA is translocated to the P site. A site is empty. So the first substages continue lah. Means the codon recognition will reoccur. Okay. So the next tRNA which occupy the A site must carry anticodon 3' UCU and carry amino acid code by codon AGA. So AGA is code for arginine. Okay, now already codon recognition done. Second is peptide bond formation. So the proline will form peptide bond with arginine. The third is translocation because now it's not the amino acid tRNA anymore. So it need to translocate to the P site, which the site for peptidyl tRNA. So here translocation occur. Okay. And then codon recognition again. So the next coming tRNA must carry anticodon GAC and carry amino acid coded by CUG, which is leucine. Settle codon recognition. Then peptide bond formation between arginine and leucine. The third one is translocation. Ribosomal subunit move one codon ahead. Okay. Now we reach at the third stages, which is termination. If you can remember, okay, I asked you to memorize 3 stop codon, okay, UAA, UGA, and UGA. Okay, so here is one of the stop codon, which is UAA. So what happened when the A site reach one of the stop codon? So the first thing is the release factor, okay, which is the tRNA with carry no amino acid. Why no amino acid? Because stop codon will not code to any amino acid. Stop codon will not code for amino acid. So release factor will bind to the A site. So when it binds to the A site, okay, what happens next? It will promote the hydrolysis of the bond between tRNA at the P site and the polypeptide. Here is the bond between the tRNA and the growing polypeptide. The release factor will catalyze the hydrolysis to break the, okay, to release the bond here. So now the polypeptide chain release. The polypeptide detach from ribosome. The tRNA also will left the ribosomal subunit. Then the ribosomal subunit will detach from mRNA strand. So now we already have the polypeptide. That is the end of the translation process. What should you know in translation? The one of important term is polyribosome. It means a multiple ribosome attached to the single mRNA strand during translation process. Okay, here, okay, it show multiple ribosome attached to the single mRNA strand during translation process. And this is important to accelerate the production of protein or to accelerate the process of protein synthesis.
Protein synthesis involves two processes, which is transcription and translation. Translation is a process using code in mRNA to produce polypeptide, and it occurs in the cytoplasm. Translation has 3 stages: initiation, elongation, and termination. During initiation stages, small ribosomal subunit will bind to the start codon, which is AUG. Next, initiated tRNA will carry complement anticodon and carry amino acid methionine will bind to the start codon. Next, large ribosomal subunit will bind to the complex, where the initiated tRNA will occupy P site. During elongation stages, it can be divided into 3 substages. The first one is codon recognition. The next coming tRNA must carry anticodon complement to the codon on A site and carry amino acid coded by the codon. Next, second substages is peptide bond formation between amino acid on P site and A site. The third substages is translocation. The ribosomal subunit will move one codon ahead. So tRNA that carry growing polypeptide on A site will translocate to the P site. A process of elongation continue. Here show the process of codon recognition, peptide bond formation, and translocation. The process of elongation will stop, and the process of termination occur when the A site reach one of the stop codon. Example here is UGA. So the release factor will bind to the A site and cause the ribosomal subunit detach from mRNA and the polypeptide release. Thank you for watching. See you in the next video. Bye.